Good afternoon, everybody. This is the CHIP Landmark Ideas Seminar. The title for today's talk is Searching for the Origin of COVID-19 by Alina Chan, a postdoctoral fellow at the Broad Institute of MIT and Harvard University. I'm Ken Mandel. I direct the Computational Health Informatics program at Boston Children's Hospital. We were founded in 1994. We're a multidisciplinary applied research and education program. You can learn more about us at chip.org. The Landmark Idea Series is an event led by CHIP and featuring thought leaders across healthcare, informatics, technology, science, and more. Dr. Chan is a molecular biologist at the Broad Institute who specializes in gene therapy and cell engineering. She uses methods to engineer viruses to cure diseases. Many postdoctoral fellows in the health and biosciences are required to take courses in the responsible conduct of research and in methods for enhancing reproducibility. It would not be an exaggeration to say that Dr. Chan developed her own independent curriculum on these topics, putting under a microscope the scientific publications and public discourse about the origins of the COVID-19 pandemic. Her tireless push on social media has brought the lab leak hypothesis for the origins of COVID, quote, from conspiracy theory to just barely a possibility to a plausible hypothesis worthy of a credible investigation. The subject has been contentious, and I imagine her postdoc years have differed sharply from what she anticipated. Tomorrow, HarperCollins will publish Viral, the search for the origin of COVID-19. We'll put the link into the chat now and also at the end of her talk. And today, we're very fortunate to have Dr. Chan with us here to tell us about her journey and the state of play in discovering the origins of the pandemic that just won't go away. If you're interested in tweeting, these are some handles and hashtags that you can use. Dr. Chan's, the Computational Health Informatics program, and the Landmark Ideas. And we've just done a brief intro. Now there will be a presentation and it will be followed by a Q&A. You can put your questions into the Zoom Q&A box as we go. No need to wait until the end. Use the Q&A box, please. And with that, I'm going to turn it over to Dr. Chan. Alina, it's a pleasure to have you here today. Please take it away. You're still on mute.
Oh, thanks, Ken. Sorry. I was just saying I'm super honored to be here to give this talk. I know that usually these Landmark series talks are reserved for people who've done so much with their lives and their careers in science. I am in a pretty extraordinary situation here. I'm a postdoc at the Broad, but I have just recently written a book about the search for the origin of COVID-19. And so in the next hour, I will be talking about what are the plausible origin scenarios. What is the status of the investigation into the origin of COVID-19? What do we know or know we don't know? And lastly, how can the scientific community prepare for future infectious diseases? Because there are lessons to be learned from this pandemic. So other people had more relaxing side hobbies, I feel like, during the pandemic. Like there were people who got a pandemic puppy, like people who learned how to bake, people who reconnected with nature. But for me, it was absolutely unpredictable and insane. Like my story has already been told by other journalists, like in Boston Magazine, by the MIT Tech Review, and by the New York Times. So I won't say too much about me. But how I started getting into this topic, looking into the origin of COVID-19, it really started with this preprint that I put online in May of 2020. So all this preprint said, and it was very carefully written with caveats laid out in the preprint, it just asked, how did SARS-CoV-2 adapt for humans? Because by the time it was detected in December, it seemed pretty well adapted for human infection and transmission. And I can speak more of this after the talk if people are interested. And one line in particular, and this long preprint got us into an unbelievable amount of mess. So we laid out different scenarios, including natural origin scenarios, and we said also that even the possibility that a non-genetically engineered precursor virus could have adapted to humans while being studied in a lab should be considered regardless of how likely or unlikely. We had no idea that a single line was going to result in a media like firestorm. So in fact, when I asked my friends what I should do about my preprint, they said you should put it on Twitter. You should make a tweetorial, which is like a string of tweets that acts as a tutorial for your paper, which I did, and that's how it got us into the media. So I'd say be careful when you make tutorials. So somehow I managed to make it through that whole year and a half, and in early 2021 this year, in March and May, I was really pleased to co-organize and co-sign letters pushing for an investigation into both natural and lab origins. So investigating both objectively and scientifically. So it brought me to this point as well, where I've just finished writing a book and it's releasing tomorrow. It has all of the details of all of the findings and key figures that I learned about on the way. And the book doesn't force an opinion on anybody. It lays out the arguments for both a natural and a lab origin. And I know that my co-author and I, Matt, really, we do lean towards a lab leak based on all of the things we've seen. We do lean towards a lab leak, but in the book, as best as we can, we try to lay out what we know to solidly be true, just based on facts. And so I hope that people will read it and let me know. I know that people either hate or love this book. I'm sure people who believe strongly in the natural origin of COVID-19, there's nothing that will persuade you. So, but I still hope you give the book a chance and take a read. I'm okay with flames. Okay. So whose stories and discoveries am I talking about today? Because there's no way that I, a single postdoc, like found out all of this information. So there are three groups. One is scientists. Believe it or not, there are many top scientists and regular scientists like me who have spent a lot of time pursuing facts and trying to figure out what happened here, what happened in Wuhan, how did this pandemic get started, and just a huge body of journalists from all around the world. I have acknowledgements at the end of this talk, but just people relentlessly trying to investigate, trying to hear the truth of what was happening, and sleuths. So people, independent analysts, people at home like teachers, bankers, like data analysts, they didn't let their lack of a scientific education stop them, and they managed to make major contributions to this story, which I will be telling today. And so these are all experts and investigators who really did not accept how a lab origin of COVID-19 had been cast as a conspiracy theory, were willing to be seen as conspiracy theorists while doggedly trying to find as many facts as possible. So we should care where COVID-19 came from. So before I tell you what we know about where it came from or not, it's kind of surprising that in the past 2 years, it began with, it came from a wet market. It started with all these experts telling us, yeah, we know it came from a wet market. And now we're at a point where some experts are saying maybe it came from a lab, but the origin actually doesn't matter. So I'm kind of surprised that this is starting to trend, that people are starting to say it doesn't matter where this pandemic has infected hundreds of millions of people came from. I'd say that it matters for three reasons, right? So one is strategy. So the details about how this pandemic started are required to inform solutions, preventions. So how we stop the next pandemic from happening, we need to know the details, the nitty-gritty stuff about how this virus got its beginnings in Wuhan city, how it started to spread. So without that kind of detail, it's really hard to formulate like strategic decisions. The second is to set a precedent. So we need to show that we can make best efforts as scientists, as investigators, to track the origins of any novel outbreak. If we show that there's no way we can track outbreaks of ambiguous origin, it sets a really bad precedent for the future. And lastly, it's really about human resolution. So we've all been affected in some way by this pandemic. I think a lot of people want to know why this has happened. And this is something that I hear a lot from people sending messages to me, is that they want to know why this is happening. So it's a human answer. So what are the possible ways that COVID-19 could have emerged? And I'd say there are two groups. One is a natural origin and one is research related. So natural origin wise, it's a story we've heard like thousands of times. There's a person who goes to a wet market or wildlife market and is exposed to an intermediate host. So an animal that is not a bat, but is more similar to human beings. So something like a civet cat or a mink or raccoon dog. Uh, and this really can come in the form of many things. So not just the wet markets, but the farms, the trafficking process, people invading habitats, causing these animals to be interacting with people. Or you can have a direct exposure to the natural reservoir of some of these viruses, like SARS-like viruses. Their natural reservoir are bats. So bats are typically hiding in these remote caves or rural areas at best. And if you are a miner or a villager, you will have exposure to these viruses. So if you're exposed to these thousands of bats every year, you might get unlucky and eventually catch something that could see the outbreak. So how about research related? And these are not on opposite spectrums. So if you look at this arm of research related origins, it could be scientists sampling animals or people. And we know that this was ongoing and is still ongoing, where scientists from numerous countries are going out there intentionally looking for animals or people in the wildlife trade, farming wild animals and taking pathogen samples from them. So tens of thousands of pathogen samples taken from animals and people in high risk occupations. So during this process, a scientist might get infected regardless of how much PPE, protective personal equipment you're wearing, or just by accident when the situation gets out of control and animal scratches or bites you, you know that it's out of control. So these are similar to natural exposures, but even the US intelligence community calls this type of origin scenario a research related scenario. And I think the answer for that is because in order to prevent this route of emergence, you have to regulate the research activities. You're not regulating the wildlife trade activities. So the last one, the most extreme lab origin scenario is where the virus has been taken to a lab. Experiments are being done with it. You're growing it up, maybe synthesizing it, recombining it, making chimeras or genetically engineering it. And you are testing these viruses on cells in the lab, different cells, animals, human cells, lung cells, testing in a variety of animal models, as we'll see. And during that process, it accidentally escapes and causes an outbreak. So I think let's get the really tough topic on the table now. So people are most worried, I think, about a genetically engineered origin of SARS-CoV-2. And the reason for that is that there is this one feature, you might have heard of it already. It's called the Furin cleavage site insertion. It's in the spike of a SARS-2 virus. So the SARS-2 virus has all of these spikes. So this is a trimer sticking out of its surface. And the open conformation, there's a receptor binding domain that sticks out, and this binds to the ACE2 entry receptor on host cells. So if you cleave at this furin cleavage site and the spike, you will favor the open conformation. So having this furin cleavage site, there's just been dozens of studies, and many experts have said that this site is what makes SARS-2 a pandemic pathogen. So without this site, when they study mutants that they've removed this furin cleavage site, it becomes completely attenuated. So it's very unlikely that such a virus will cause a pandemic. And if you look at all of the SARS-like viruses, so here I have SARS-CoV-2 represented by Wuhan-Hu1. So this is one of the first human isolate sequences that was released online of SARS-CoV-2. You'll see that it's the only one with such a unique furin cleavage site insertion. So all of the rest in this SARS-like virus family, they don't have such a furin cleavage site. And it's unclear whether they have similar insertions in this area. It's a very structurally flexible loop. So for a deeper perspective, I recommend reading the paper that my colleague and I just got through peer review. It's in Molecular Biology and Evolution. So we do a really deep dive on this topic. So again, if you look at all of the most closely related sequences, viral sequences, this furin cleavage site insertion sticks out like a sore thumb in SARS-CoV-2. So it had to have emerged pretty recently to have this difference compared to all of its relatives with highly conserved flanking sequences. So the problem is that this pandemic had only just begun when some scientists and journalists started to call the lab leak hypothesis a conspiracy theory, a racist theory, or an anti-Chinese theory. I feel like these assertions, or the light that was cast on it was very unscientific. And I'll show you some examples, but they are not the only ones. So for example, there's this famous or infamous Lancet letter that was printed in February of 2020. So barely a month since the genome of the virus had been put online. They condemned all conspiracy theories suggesting that this virus doesn't have a natural origin. They claimed that scientists had overwhelmingly concluded the natural origin, and they put behind them the US National Academies of Science, Engineering, and Medicine, and the scientific communities they represent. So it basically, I think, had a very strong chilling effect, or it scared people away, scared other scientists from saying that they might think this could come from a lab. Another really highly cited paper or opinion piece that came out in Nature Medicine in March was written by 5 really well-known virologists and evolutionary biologists. They said that we do not believe any type of lab-based scenario is plausible. And in a press release that went out of that, one of them went even further to say that they could rule out all lab-based scenarios. In May, even by mid 2020, this was still ongoing. So the Center Infectious Diseases Research and I think Progress, they said that even a lab release, and like accidental release of a natural virus, was a conspiracy theory. So can you imagine all of these top scientists and top research organizations calling one plausible scenario a conspiracy theory? So it's really, really difficult for anyone to say that this virus needs to be investigated, whether it came from nature or from a lab. All the major social media platforms, except for Twitter, started to ban these things, to shut down and censor people even raising it. So it was not a good situation. It wasn't conducive to objective scientific exchange. So the issue is that today, in like 2019 and onwards, I mean, actually since 2016, scientists have had the technology to synthesize virus genomes seamlessly without leaving traces of engineering. So if you talk to the experts in this field, for example, Ralph Barrick, who worked very closely with the Wuhan Institute of Virology to engineer chimeric synthetic viruses, he said you can chemically synthesize the sequence of the virus in the lab. The answer will only be within the records of the WAV, whether they had it, whether they had engineered this virus, right? You asked Jack Nunberg, a virologist at U of Montana, who himself had inserted a furin cleavage site into the spike of SARS-1, and he said there's no way to know whether humans or nature inserted the site. Gary Whittaker, Cornell, he is also a virologist who inserted an FCS into the spike of SARS-1. And he said this motif in SARS-2 is highly unusual, leading to smoking gun hypothesis of manipulation. And David Baltimore, another extremely renowned virologist, he was the one who actually said the smoking gun hypothesis first. He says it's really a question of history, because you can't look at the sequence alone and distinguish between the two origins. So if you talk to the top experts who have themselves even engineered or inserted furin cleavage sites, they all tell you there's no way to know. You can't just use machine learning to tell you whether this has been tampered with in the lab. There have been also many key precedents of lab-based outbreaks. And if you read the book, we go into all of these in detail. I can't go into all of them because of time constraints. But let's see, okay, in 1967, a completely novel virus, the Marburg virus, exploded in 3 labs, causing outbreaks. So at that time, no one had even known about this virus. So the first time it emerged and was known by humans, it was from lab. It was a lab-based outbreak in 3 different places where monkeys have been shipped. A whole pandemic has been the result of failed vaccine trials, so research activity gone wrong. That was in 1977. In 1979, anthrax leak happened. Dozens died after a single air filter wasn't replaced in this military lab. There was a major cover-up. People only found out decades later. Actually, people only found out about this pandemic also decades later, the source. It sometimes takes many, many years before someone who knows something feels like it's safe enough to come on and say, actually, this happened. 2004, the SARS virus, the first SARS virus, when it was studied in labs, escaped at least 6 times from labs in 3 countries. every time it escaped, there was a surprise to the people and the scientists that they were completely surprised that this could happen. In many of the cases, they couldn't even identify when the accident happened, when the exposure had happened. They didn't know. It was only retrospective. Once it had infected someone, then they realized, oh, it escaped. One of these labs was a top biosafety lab, a BSL-4 lab, and it still escaped. So in 2019 itself, there was an outbreak of brucellosis. This is actually a bacteria. Like thousands of people infected after expired disinfectant was used. So it leaked from lab. So it's very normal to have pathogens escaping from labs nowadays, especially considering the dozens of labs being built and more scientists and researchers getting engaged in this type of work where you're working with animal or human pathogens. So in 2019 alone in the US, per week, there were on average, more than 4 accidental releases of select agents. So select agents talk about the most dangerous pathogens and toxins. So this is happening at a frequency that I think a lot of people are not aware of. And you only need one really unfortunate time to see a pandemic if you're not careful. So what is the status of investigations into the origin? Where are we at right now? We had a flawed China WHO study that even their leaders and the WHO insist was not an investigation because they had no mandate and no power. Basically, what they did was they went to China, they discussed the analysis of Chinese scientists, and then they signed off on the report. And this report, it said that they thought an introduction, so the pandemic could have started through cold food chain products, like frozen products imported into Wuhan. They thought this was a possible pathway, but they ruled out an introduction through a lab incident. So they preferred the emergent scenario where it has nothing to do with China compared to a very reasonable origin scenario where an accident happened. Okay. So in response to that, a lot of scientists were pretty shocked to see the World Health Organization sign off on a report like that. So I was part of this team. When we saw that report from the WHO team, we said, as scientists, we have to say something about this. This is just bad. That's just bad for the scientific community, for public trust in science. So we wrote this letter asking for an investigation into both natural and lab origins. So we still want to investigate both origins objectively in a science-based manner. This story had a huge impact. It was covered by like 618 outlets, 1,700 news stories, and I heard it impacted and led to this White House statement by President Biden, who called the intelligence community to investigate the origin of COVID-19. So he asked them to get all of their intel together and put out a report. So this was in May, and the report, the short declassified version, was released in August. So what did it say? It said, oh, sorry. So just before that, the IC is actually made of numerous member agencies, and some of them are tasked to do this, to do each inquiry, but they don't necessarily share all of their intel with each other, and they don't necessarily all interact with each other. So what happens is that each one of them that are involved in a specific inquiry will come up with their own vote or debate process and say what they think is most likely or not. So what happened is that most of them were in the I don't know area or in the low confidence area. So not enough evidence of low quality. At least 4 agencies said that they think it's most likely it's natural, but with very low confidence. Only 1 agency was outside of the low range and voted for a research-related spillover. So according to the New York Times, this was the FBI. But really, if you look at the whole thing, what this really means is that people are really undecided and they're not necessarily sharing information with each other. So there's really a lot more work to be done here. This cannot be the end of an investigation. There's much more to be done. So ultimately, all agencies assess that the 2 hypotheses, both natural exposure and lab related, are plausible. One thing that was kind of surprising to me is that some analysts on the side of a natural origin gave weight to it because they said that the Chinese officials were not surprised. They said that the Chinese officials were surprised by the pandemic. But my point is that when a pathogen escapes from a lab, everyone is surprised. Even the scientists themselves are surprised. So especially with something like SARS-CoV-2, where it's a very sneaky virus, it causes a whole spectrum of different symptoms. You may not know that you were even infected. It would be a while after, like several weeks after an incubation time of 2 weeks per patient, that you might detect actually that there are people who have been infected by this virus. So I don't think that this reasoning is correct. That it's not that when someone is infected in the lab, they suddenly start growing, glowing green and purple, and a light shoots up into the sky to let everyone know that they've been infected. So I think they need to reconsider this point. So right now, what's happening is that we have a COVID commission planning group going, and they are planning a commission. So they're not actually a commission, but they're trying to plan one. And it's funded by many private donors who are very interested in solving this problem and making sure that the country is prepared for a future pandemic. We also have a bipartisan bill right now by senators on both Democratic and Republican sides, and they want to create a 9/11 style commission. So I'm very encouraged to see that these are in the pipeline. So what do we know or know we don't know? So this is the more interesting part for me as a scientist. So surprisingly, we don't know when the outbreak began. So the China WHO joint study did not succeed in identifying cases before December, which is very surprising to me because we know that that was tip of the iceberg. We know that there were possibly thousands of cases by that point. There's no genetic or epidemiological evidence for extended period of pre-circulation, meaning that there's nothing to suggest that this virus had been around for a year or more. But there are some really practical action items that have reportedly still not been done 2 years later. So imagine you're a local government or the Beijing CDC, and a new SARS virus has just broken out in your town. Doesn't it make sense that you would at least try to contact trace the earliest cases to see what the exposures were? So apparently this has still not been done, which is pretty crazy. They've said that they're now testing blood samples banked in 2019 because they couldn't get consent. So now they say that 2 years has passed, so they can start testing blood samples that were banked in October of 2019. So if you look at the data describing the earliest cases, you see that many of the first cases had no exposure to a live animal market, no exposure. And if you look at the case severity, you could see a lot of very severe cases, ICU cases, even in a young range, like 25 to 49. So I bet what they were seeing, and I think many experts will agree, that they were not seeing the bulk of cases. They were seeing the most severe cases. On the side, it's been leaked that the USIC, the intelligence community, has data relating to 3 Wuhan Institute of Virology scientists who had worked with bat coronaviruses who went to the hospital with COVID-like symptoms in the fall of 2019, but these details are still classified. I've heard that people are trying to push for those information to be declassified, but it would be very interesting to look at. So how do we know that the outbreak was probably much larger than reported? So let's just look at the population density in Wuhan city. So if you look at the city, especially in central Wuhan, this is where all the population is concentrated. And they have an aging population, which is pretty typical of a lot of Asian cities. So if you look at where all the elderly population are populated, they're all here in central Wuhan city. And if you look at the case information for the COVID cases that were detected, you see that they all cluster at the most densely populated with the most elderly in the city. So it makes complete sense that when you have detected an outbreak, you're detecting the most severe cases in the most densely populated part of town. The problem with the data that we have about the earliest COVID cases is that it actually comes from this extremely low resolution picture from the China WHO report. So the authors of this cell review, what they did was to use Adobe Illustrator Pathfinder to reverse engineer this picture and get this. So it's really not good for science that this is happening, that we have no access to data, and people are using Pathfinder and Adobe Illustrator to make figures look nice like this. And we even know from the WHO China report that this map is wrong. Like, their report contradicts their map, because they found that actually the first case of COVID-19 that they know of he went to a market here right next to the Wuhan Institute of Virology building in Changsha, but they didn't map him onto the map. So there's something very wrong with this map. We have no access to the data. So I'd say that this is a pretty bad attempt at contact tracing and mapping out the earliest cases. All right, next topic. So was the seafood market actually the site of a natural spillover, or was it a human super spreader event that happened later? So many of the early cases, including the first patient, had zero exposure to the market. As early as May 2020, the Chinese CDC director himself said actually none of the animal samples they took from the market had any signs of virus. They said at first they assumed the seafood market had been the site of natural spillover because it looked very similar to SARS-1, right? But now they realized that the market is more like a victim. The novel virus had existed long before. And okay, you don't want to believe the Chinese CDC director? Okay, but if you look at the genetic data that the whole world has seen together, and I really have to credit Jesse Bloom here from Fred Hutchinson, who recovered data that was deleted off public databases. And he found that if you look at the earliest variants of SARS-2, so I can't really explain this because I don't have enough time, but you should go read his paper. It's really good. You see that this seafood market only had variants in one lineage. The other early variants didn't present at this market. And we know that the earliest cases didn't have any links to this market or any market in any live animal market in town. So it is most likely, based on this data, that the market was a later cluster of the virus. It had pre-existed the market in Wuhan city. So where is the proximal animal source? Again, the Chinese CDC director said that they haven't found any animals from the market. In February and March of this year, the China WHO joint study said that they went through 80,000 animal samples, more than half were from wild animal species, found nothing. The Wall Street Journal reported that farms in South China that had supplied that Wuhan market was shut down without testing. In early 2020, all the animals were bought and they shut down without testing. So that's very strange because even if you want to shut down the farm, don't you want to test it first just to definitively know whether it was the source of the virus? So we're kind of at the dead end here. But in June of this year, a paper that had been languishing behind peer review, like it's kind of hilarious how this is happening, that key data relating to the origin of a pandemic is hidden behind peer review. Like the editors wouldn't publish it, didn't consider the timeliness of releasing this data. Finally, data showed that there were actually some wild animals being sold in Wuhan between 2017 and 2019, although the raw data has not been made available. So we don't actually know how many of each animals was sold in the fall of 2019. The authors had done this to study a different tick-borne disease. And back then, nobody thought that nobody knew that pangolins would come under suspicion. So the people selling these animals in the market had shown them everything. There was no reason for them to be hiding bats or pangolins in particular. Like they didn't predict that pandemic would be right on their doorsteps. So there were 7 out of 120 shops in Huanan that sold these live animals, but these numbers represent all of the markets in Wuhan that were selling these animals. So you see, for example, a palm civet, which might be a potential intermediate host. Only about 11 of them were sold per month with a standard deviation of 8. So some months there might have been like just 1 or 2 being sold. And I was told by the authors of this study that the sales would go down at the end of every year. Starting in November, there would just be very little sales of these animals. It was a seasonal thing. So the problem is, why haven't any of these suppliers been tracked and tested? You'd think that if a killer virus emerged on your doorstep, you would feel some sense of urgency of tracking the suppliers of these wild animals. And this is where the China WHO study, again, makes zero sense. So that study, when they came back, the WHO team told us that they were told that no live wild animals had been sold at this market. And so in this map that they showed us, these dashed boxes are just shops that sell domesticated wildlife products. So products, not actual live animals. So they showed these, and then they mapped out where all the environmental samples had tested either negative, like blue, or positive for SARS-2 material, genetic material. because again, 0 animal samples had shown any signs of the virus. So all they had was environmental samples like sewage, door handles, like chopping boards, that kind of thing. And they showed the early cases as well, so shown by the red dots. So if you look at this map, even just places selling wildlife products, not even the animals, they don't particularly map onto where you actually get positive environmental samples. Even the cases don't map onto where these shops selling the wildlife products are. So this is absolute misinformation that people have been spreading. I've heard it even from other scientists claiming that the early market cases were linked to wildlife products. There's no data that substantiates that claim. And also important to note, this market was located in central Wuhan, again, at the intersection of major metro lines, like millions of people taking that metro line. It was humongous. 120 shops, 50,000 meters square of retail floor space, 10,000 visitors a day. And it's not mind-blowing that there would be a human super spreader event there. In fact, super spreader events of COVID-19 have occurred in markets in many other countries. It doesn't mean that there was always a sick batch of civet cats. So even more recently, just like last week, a study went on bioRxiv showing that they had done a pretty exhaustive study between 2017 and 2021, sampling 344 locations across 19 Chinese provinces, close to 2,600 samples, 1,700 animals, and 16 species, and found 0 SARS or SARS-like viruses in the wildlife trade in China. So I'd say that right now we have like no evidence of any of these SARS-2-like viruses being passed around the wildlife trade. There's no sign of SARS-2-like viruses in the animals in Wuhan. So what does this mean, right? So it means that up till now, despite all these searching across 8 countries in South China and Southeast Asia, the only SARS-2-like viruses found in association with wildlife trade were 3 pangolin viruses, a total of 3 pangolin viruses. And we now know that there were no pangolins in Wuhan city. If you look at where all the closest relatives to SARS-2, again represented by Wuhan here, they're all from bats. So they're pretty spread out across South China, Cambodia, Laos, Thailand, in that hot spillover zone, but not in central China, right? But they're all from bats. So you have to go into these very remote caves and mines to find these very close relatives to SARS-2. A bit further out, you see a few, but they're all from pangolins. So there's no sign of these viruses in like civet cats or raccoon dogs, so the animals you'd normally expect to be sold at a wildlife market in Wuhan. So one other question that I've seen a lot is people say, is it normal for virus to be all over the surfaces in a place where people have been sick? And the answer is yes, it's very normal. And that's why in the early phase of the pandemic, and even now, there's all this hygiene theater, like people are spraying and cleaning everything like crazy, even though we know this virus spreads through the air. So virus on surfaces in a place doesn't mean that there is a sick raccoon dog somewhere. For example, the Diamond Princess cruise in February 2020, when the cleaning crew boarded the ship, they found just virus material all over the cabins of both symptomatic and asymptomatic passengers, which made them really concerned about fomite, the surfaces transmission, people touching their faces and surfaces and then catching the virus. Nation News covered a series of studies checking how long the virus could remain infectious on different surfaces, and it could be for days. So on plastic, steel, very normal surfaces to encounter in your workplace, on the market. On banknotes, it could last for 3 days, like surgical masks, 7 days, and on leather for 8 days. So it's not surprising at all that if you have lots of sick people around, the whole place is going to be covered with viruses in the air and on the surfaces. So it doesn't mean that there is natural spillover actively going. Like there wasn't like a whole batch of mink on this Diamond Princess cruise. It's not that kind of boat. All right. So then the question is it usually this difficult to track down the proximal animal source? And the answer is no. So there's been another batch of misinformation floating around where people have said it takes like a decade to track the proximal animal source. That's not true. And I think they're very misinformed. So just take a look at SARS and MERS. All right. So SARS-CoV, it was so rapidly determined, the zoonotic origin. So as soon as they started searching, so let's turn back time to 2003. In March of that year, they isolated the virus and saw it was a coronavirus. By May, they had found a whole variety of animals in the local markets that were infected with SARS-1 virus or had antibodies to it. And not only that, but they took thousands of samples from all sorts of different populations of people in the city. And they found that animal traders, despite having not been diagnosed with pneumonia or SARS, they were highly positive for antibodies against SARS viruses. So these all were found within 2 months. Like they immediately knew that there was a lot of SARS-like coronaviruses circulating in the markets, in the wildlife trade in Guangdong, where SARS-1 burst out. By the time SARS-1 emerged again at the end of 2003, one of the index patients was a waitress. And the doctors asked her, "Do you have palm civets in your restaurant?" She said no. They didn't believe her. They just went to the restaurant anyway and took samples, and they found cages, right? So they took samples from the cages, from the animals, from all her co-workers. It was just everyone SARS. So that one took days. So nothing, no like 10-year period waiting to find the natural like proximal animal source. That's crazy. And for MERS, we now know that it has been spilling over dozens of times, like some experts say hundreds of times, from camels to humans. And if you go to camel stables and whenever there's a new MERS case presenting, like most of the time you can directly find the animal that gave it to you, or the human that passed it to you that was exposed to a camel. So it's very standard. And the thing is, this COVID-19 pandemic began in a city with the world's foremost expertise in SARS coronavirus outbreak tracking. Like the world's like foremost expert on this, Dr. Shi Cheng-Li at the Wuhan Institute of Virology, worked in that city. And even her collaborator, Dr. Lin Fao Wang from Singapore, who is another foremost expert in tracking SARS virus outbreaks, was in the city at the time as well in January 2020. So they should have had everything at their fingertips, like all the technology, all the know-how. Just they had the best chance possible to track this SARS virus, is what I'm saying. And Shi Cheng-Li said she told a reporter at who wrote the story for Scientific American, when she heard about this SARS-like virus breaking out in Wuhan, she had never expected this kind of thing to happen in central China, because all of her studies, and I'll touch on some of these briefly, have shown that it's only the southern Chinese provinces and Southeast Asia that have these SARS-like viruses that jump from animals to humans, particularly from bats in those provinces. So she said, if coronaviruses were the culprit, could they have come from our lab? So for her, she thought that it was unexpected for this type of viruses to come out of the natural wildlife trade. She thought, could they have come from my lab? And I thought this line by Rowan Jacobson, a journalist writing for Newsweek, was very insightful. So despite 15 years of coronavirus hunting and testing by the Wuhan Institute of Virology, it was helpless to prevent a pandemic of that same virus in its own backyard. So if this is a coincidence, it's one of the great ironies of history. I'd say it's a cruel irony that this would happen to them. So, okay, we went through some of the data for natural origin. What about lab origin? So what kind of research was conducted? What do we know about the type of research conducted in Wuhan using SARS-like viruses? So we know from the documents that have been slowly extracted from one of their longtime funders and collaborators, a New York-based organization called the EcoHealth Alliance. So their first aim was to characterize all of these high-risk pandemic potential SARS-like viruses in South China and Southeast Asia. So collecting all of these animal samples from bats and likely hosts in the wildlife trade and sequencing them. They were surveilling spillover into humans. So they were actually collecting thousands of samples from humans in the wildlife trade or in hospitals who were presenting with unresolved illnesses. And lastly, once they had these novel SARS-like viruses, they were testing their spikes in pseudoviruses or recombinant viruses to infect different human cells, different human ACE2 expressing cells, as well as animal models. So we know that they had experimented with humanized mice, civets, and bats with SARS-like viruses. So most troublingly, a DARPA proposal was leaked, and this proposal was submitted in March 2018, which included a roadmap that the scientists were looking for and inserting novel cleavage sites into novel SARS-like viruses for infection studies in the lab. So this is very disturbing, and I'm going to get into it. So from one of the Freedom of Information Act documents by The Intercept, this shows the EcoHealth grant that was done in collaboration with the WIV. In fact, all these experiments were done in Wuhan by WIV scientists. So they made chimeric SARS-like viruses, and unexpectedly, one of these, or actually a few of them, showed gain of function in humanized mice. So why were they doing this experiment? They were trying to learn more about natural SARS-like viruses and figure out which ones pose a threat to humans, which ones could cause disease in humans. So for them, they had to develop models to study how novel SARS viruses cause disease. They said that having these humanized mice, in this case, mice expressing the human ACE2 viral entry receptor for SARS-1 and SARS-2, it was valuable as a model for assessing the pathogenicity, the ability to cause disease by novel SARS viruses in these animals. So why were they doing this? Didn't they realize the risk of generating a pandemic potential pathogen? So if we look at what is published in the scientific literature from 2017, they were already making these chimera, but they did not exhibit gain of function in human cells. So if you look at the experiments in human cells, and I don't have time to get into this, but you can definitely find the paper online here, PLOS Pathogens 2017. None of the chimeric viruses, so what they did was they took a WIV1, the first recorded SARS-like virus that they found and isolated in 2013, and they put in the spikes of other novel SARS viruses they found in nature. And none of them performed better than the parent, regardless of the cell that we're using. So they were using human cells with human ACE2 expression. But once they moved into humanized mice, that changed completely. So they unexpectedly showed a strong gain of function in humanized mice. So you'll see that the parent is this WIV1, and these are the chimera. So some of them could cause more severe disease, more body loss in these humanized mice, and troublingly, like massively amplified viral loads in the lung tissues of the mice infected with these viruses. So this is the parent, WIV1, and in some cases you can see that there's like 10,000 fold higher viral loads in the lung tissues. Their wording, not mine. So they also they continue doing these experiments with the chimeric SARS viruses, and they found that this also had a tremendous impact on viral loads in the brains of the infected human ACE2 mice. So the parent is not even visible on this graph, but you can see that this is log, by the way, log genome copies, which they use to estimate viral load. So some of them would just massively gain of function compared to the parent. And at least one of them caused a very significant enhanced mortality in these humanized mice, so causing most of the mice to die compared to the parent here, the WIV1 in black. So these were all unpublished data that had to be Freedom of Information Acted out of the NIH, unfortunately. This information wasn't shared with us when SARS-CoV-2 emerged. And troublingly, from the same batch that was only just released in this fall, this is data they had from 2019, before the pandemic. They were doing the same experiments, chimeric MERS-CoV, using human MERS, not bad MERS viruses. They were using human MERS, the one that kills like a third of the people it infects. They were switching in and out spike receptor binding domains from MERS-like bad viruses they had found in China. So all these chimera. So in human cells, again, they didn't see any gain of function. But what happens when you put it in humanized mice? So we know that it was in their plans to do so. We only have data from 2019. So what happened afterwards? Did they actually follow through and test these viruses on humanized mice? And what was the biosafety level they were working with? So it's very troubling to hear that from their publications, from their own publications, they say they were working with these live viruses and recombinant viruses under biosafety level 2 conditions. This is from a thesis by the same student in 2017. And when Shi Zhengli herself was interviewed in July 2020, she said, all the coronavirus research in our lab is conducted in BSL 2 or 3 because they were working with bad viruses. So according to their national or local regulations, this work could be done in 2 or 3. It didn't require a high biosafety level. They found that they could infect civets directly. So they were experimenting with civets in the lab too, with these bad SARS-like viruses. It showed low pathogenicity in the mice and no pathogenicity in civets. These data is still being sorted and will be published soon. That was told to us in July 2020. I personally have not seen where it has been published. Maybe it's in peer review somewhere. And if it is in peer review somewhere, I'm asking the journal editor, can you please speed it up a bit? Because it's pretty important to understanding what research was being done in Wuhan. again, more leaked documents leaked 2 years after, close to 2 years after the pandemic started. This is the DARPA proposal from March 2018 submitted by the EcoHealth Alliance in collaboration with multiple labs in Wuhan as well as in the US. So multiple people were aware of this grant, said nothing when SARS-2 emerged. So we will introduce human-specific cleavage sites and evaluate growth potential of these SARS-like viruses in viral cells and human airway epithelial cultures. We will review our deep sequence data. So who had access to all these sequencing data, right? For low abundance, high risk SARS-CoV, so rare but dangerous SARS viruses that encode functional proteolytic cleavage sites. And if so, if they find these sites, they will introduce these changes into the appropriate high abundance, easy to grow in the lab, but low risk attenuated parental strains, SARS viruses. So they had a roadmap. Up till this day, SARS-2 is the only SARS-like virus that we know has a furin cleavage site insertion in the spike and the S1, S2 region. Did this group of scientists find rare cleavage sites in their sequence data? And what did they do with that? This is from March 2018. So there's a good year and a half before the pandemic started. Like, we really need to get a hold of those communications and documents and figure out how this paragraph came to be in this DARPA proposal. Although it wasn't funded, this was a very highly funded group of scientists. They had hundreds of millions of dollars amongst them to do this type of work. So also here, they said that they would synthesize consensus candidates of genomes. So even if they couldn't isolate the virus from samples, they could just synthesize the entire genome. And they were doing this seamlessly since 2016. So they had the technology. There's no need to put your signature or sign your name in the virus. You can just make it look incredibly natural. In fact, when SARS-2, its genome was uploaded online in January, it took researchers in Europe and in the US separately. They each took only 2 to 3 weeks to completely synthesize the SARS-2 genome, making it look exactly like the parent. In fact, the researchers had to deliberately put in mutations to discern which was the virus from nature versus the virus they created in the lab. So I think this is very problematic. Do we know what viruses and sequences the WIV has? And the answer is no, because they had a giant specimen database specifically for looking for animal and human pathogens that was taken offline in September 2019. For whatever reason that is, I don't care, but they haven't put it back online. They haven't shared this database, which we know can exist as an Excel sheet, with anybody. So even though they had raised so many millions of dollars to conduct this work and to create this database to predict and to inform the response to pandemics, when the pandemic actually happened, they didn't share the data, which is crazy. Pandemic database not available when pandemic happens. So just speechless. All right, so, and if you look at the literature, the published literature, we can see with our eyes, and this analysis is all based on open source data. So anyone here, if you don't believe me, go ahead and redo the work yourself. This analysis was done by Francisco de Ribera. He went through all of the papers from the WIV and EcoHealth and pieced together all the IDs and the collection times. And he found that we see this growing number of viruses published, collected up to like 2015, a few in 2016. But that means that it doesn't mean that suddenly the WIV gave up on discovering any more viruses. It just means that, like regular scientists, they're building their work. So they haven't published everything immediately. It's not like you get something on a sequencer and it goes into NCBI like naturally, automatically by itself. So they're probably studying stuff and they haven't shared with us what they have. So it's very troubling to me that this 3 years here, we don't know what they found in nature. And from more communications from the EcoHealth Alliance, Foyet released like last month, this is from the EcoHealth Alliance, the president himself. He says that there's a reduced amount of wildlife in South China, reduced amount of wildlife being sold. So now we have to go to 8 countries, to 7 other countries outside of China and get wildlife from them. So they propose collecting thousands of animal samples in the wildlife trade, spanning 7 other countries outside of China, and shipping all of these directly to the Wuhan Institute of Virology. This was from 2016. So again, just go back to this. We have no idea what they collected. So why hasn't this data been shared with us? Is there some future pandemic that we're saving this data for? Like, why hasn't this been made public? It actually was funded with US dollars too. So I think we have a right to ask for that information. All right, so what we have right now is that when news emerged of a novel SARS-like virus with a novel cleavage site causing an outbreak in Wuhan city, scientists in the know didn't tell us that they were working with 9 of its closest relatives, and I can talk more of this after the talk if you're interested. These were linked to mysterious pneumonia cases. They had also been collecting thousands of unpublished high-risk samples from animals and humans across 8 countries, not just China. With a clear roadmap for synthesizing consensus genomes and inserting novel cleavage sites into novel viruses, then all these animal models at their disposal, they had no hesitance to test even the most dangerous ones like human MERS coronavirus in these animals in the lab at lower than expected biosafety levels. Their database, funded with hundreds of millions of international dollars to inform pandemic prediction and response, remains inaccessible close to 2 years post-outbreak. So this all looks incredibly smoky. But I'll still say that there's still no dispositive evidence for any origin scenario. Where we are at today is that both natural and lab origin hypotheses are still on the table, and each must still be rigorously investigated. Because when you have no dispositive evidence, that means at any point, a single piece of dispositive evidence can change everything. So that's why we need to take both scenarios seriously. You cannot be guessing which one is more likely or less likely. Now, it doesn't matter. We should be focusing on getting information, on investigating. So, and even if SARS-2 is 100% natural, like not even someone who got infected was sampling animals or humans in the field, the state of pathogen research globally is very concerning. There's no evidence that SARS-2 came from that mine where the 9 closest relatives were collected and studied at the Wuhan Institute of Virology. There's no evidence from specific public domain EcoHealth and WIV experiments that SARS-2 was a product of these, but these revelations indicate a severe lack of transparency and forthcomingness from these scientists who should have been protecting us against a pandemic like they said they were doing. What they were trying to do was to learn as much as possible about pathogens so that they could inform us to the maximum ability when novel outbreaks occur of those pathogens. But we've seen the complete opposite of that, like key information that's been withheld time and again. The scientists who did this work called the lab origin a conspiracy theory, and yet time and time again have been shown to be very misinformed about the work they were doing. Like every time they said they weren't doing something, later someone would find evidence showing that they were doing that something. So the last part of the talk today is really about how scientists, us, should prepare for the next outbreak. So one is we just have to investigate. We have to set a precedent. We have to find out how we can have detailed solutions to track outbreaks. So there are many leads and sources to obtain key information from. We shouldn't give up. And again, many of these documents and exchanges were sent to parties outside of China. So we should take a look at those before we give up. That's not the scientific way. We need to start making pathogen research more transparent and safe. So I say that we need a new moratorium. Many other top scientists also say we need a new moratorium regardless of where COVID-19 came from. We need a new framework and a new review process, one that's not controlled by people who want to do the work. So you can't have people approving their own work. So, all right. And the last one which I'll actually show some slides on is to strengthen research integrity. So I think that the current publication system has so many weaknesses that they can be exploited by bad actors or sloppy actors during a time of crisis. And we need to close up those gaps before the next outbreak happens. So data sharing practices, peer review practices, reproducibility, and competing interests. So I'll quickly go through this. And okay, I have a bit more time. All right. We don't need to wait for the next outbreak of ambiguous origin to do these things. So the most basic question is, when the paper is published, does the data even exist? So apparently there's no systematic way to do that journals are checking if data even exists. And one example, but they're not the only ones, is this PLOS pathogens pangolin paper. The editor later, when he was contacted in 2020, confirmed that he didn't even know if the sample existed. Concerns about this study and another pangolin paper only came to light after. They just didn't know anything about where the data were. And they spent another 6 months after that investigating. Is the data reproducible and accurate? what is the peer review process like? So this is another egregious example, but not by itself. This paper was received by journal October 28, accepted October 29. So I know that we're in a pandemic and peer review is fast, but this is a bit too fast. Like, can we see the peer review? And so I think peer review should be open in a pandemic. And when errors are found, they have to have a quick investigation and quick correction. So publish the papers fast, yes, because of the pandemic, but quickly correct. Otherwise, it has incalculable and irreversible impact on media reports and the scientific literature. Not to mention when the study claims that they found SARS-2 antibodies in people in Italy in September. 14% of the people in September they studied had SARS-2 antibodies, according to this study. The corresponding author, first author, is the scientific director at the institute that hosts this journal. And then when someone called them out for the one-day peer review turnaround, they investigated it from March to June and said no further action is required, no revision is required. So are there competing interests that are not declared? So it transitions into this. And one famous example is this Lancet letter from February 2020, where the authors condemned all conspiracy theories suggesting COVID-19 doesn't have a natural origin. Many of them were affiliated with the EcoHealth Alliance. They said we declare no competing interests. 1 year later, they published a follow-up to that letter, clarifying that they were just expressing their opinion, which are neither data nor conclusions, and their declaration of interest, this much. This is absolutely insane that none of this were declared the first time around. So even more unbelievably, that the person who organized this letter, Peter Daszak, is the president of the EcoHealth Alliance, who was later appointed to be a subgroup leader of the China WHO study on the origin of COVID-19 and the chair of the Lancet group to study the origin of COVID-19. And everyone just stood by and watched this happen. Like, he went to China, he was part of this team, he signed off on the report. It's absolutely mind-blowing. So, okay, lastly, we need to facilitate transparent scientific discourse. So when there's an emerging issue, not everything is climate change where the science is clear. Like, when there are emerging issues, we need balanced scientific discourse. We shouldn't have gatekeeping, editorial gatekeeping, peer review gatekeeping, accusations of conspiracy theorizing or racism, deciding for the public what they can handle. This is the worst part. Seeing all the emails leaked from top scientists who were chatting with each other in early 2020, many of them were just saying, let's just completely shut down the lab thing because nobody can even handle the idea of a natural virus or a genetically engineered virus leaking by accident from a lab. So the first origin of COVID balanced scientist to scientist live public debate only took place in September of this year, like close to 2 years post-pandemic. And I was very honored to be here. I don't know why they invited me. It's just shocking that it took that long for a public scientific debate to happen. Sorry, concluding now. So we need to, as scientists, find the origin. It's doable, it's within reach. We can exert best efforts. We need to develop our tech. There's no other way to say it. We need to set a precedent for accountability, transparency, and research safety. We need to restore public trust in science. We will absolutely kick ourselves if the next outbreak of ambiguous origin happens and we've done nothing. And seriously, where we are at today is where we were at 2 years ago. There's been no measures taken to make research more transparent or safe in that time. Nothing as far as I can see on a national or international level. So just acknowledgements, all of the scientists, especially the ones who co-signed these letters, pushing for an investigation of both natural and lab origins. A lot of independent analysts, some of them are scientists too, but the degrees don't matter because they really contributed. They found open source like information, and just the extreme determination of a lot of journalists, many of them are actually based in China, like risking their lives and family safeties to report on these stories. So with that, I'll conclude this talk. Thanks for having me.
Thank you so much. There's an extremely active set of questions going on in the chat, and I'll try to select a couple, but first I'd like to ask you in two questions. Gain of function research to protect humanity. Is there a case to be made for gain of function research in pathogenic viruses towards towards a public health protection argument, or is there no case to be made for gain of function research, in your opinion?
I'm very moderate on this issue. So I do believe that there's some gain of function research of concern. So with pandemic potential viruses that is worthwhile, but we should do this in the most 100% transparent and safe way. So what we are seeing now is like near 0% transparency. And when there's near 0% transparency, that's when things go wrong. When there's no accountability, no awareness of what type of research is happening and where and when, that's very dangerous. So only when you know what research is happening, then you can put really good regulation and safety on. You can tell them maybe don't do it right next to an international airport, maybe do it like at certain periods of the year in places where there are no people and there's very good quarantine measures around that type of research.
From the perspective of BL4 labs in the United States, for example, even we have one in Boston. Are there lessons from your investigations and your sleuthing that we should be applying urgently here in our labs in the US?
Yes. So again, from a 2019 summary of accidental releases of select agents in the US, we know that they're on average more than 4 a week. That was in 2019. And this sort of research is only burgeoning globally, just like countries stating that they're going to build all these BSL trees and forests everywhere, all over their top cities. So we have to deal with this problem, and it's a global problem. It's not a China problem. It's even a Boston problem. So I mean, I know that calling it a problem is harsh, but when you have millions of lives at stake, it is a problem. So we have to make these all as transparent as possible. And the work is actually ongoing. So there is an ongoing NIH fund going again to the EcoHealth Alliance to do the same type of research, collecting bad viruses likely to spill over into humans and shipping them up here into North Carolina and into Massachusetts for experiments. So as a person living here, I'd kind of like to know what kind of research is happening, right? and what are the safety measures, the actual safety measures taken to make sure that it doesn't explode in the city.
Here's a question from the Q&A. Could you comment on what your experience has taught you about independent scientific thought and how it might be protected and encouraged?
It's a really tough question, and I'm not sure I'm out of the danger zone yet. I'd say that it's pretty dangerous, and it really speaks to the courage of all these journalists, citizen journalists in some cases, like whistleblowers, like scientists, top scientists who've stuck their necks out and said this problem is more important than me, more important than my career, because next time this happens, maybe I'm in my 60s or 70s, or maybe someone I love gets killed by the next pandemic of ambiguous origin. I will really kick myself for not having said something in this origin. We really need to set a precedent here. I know a lot of scientists who have spoken up on this topic. We're so tired. We're so tired of pushing for an investigation. And we have to acknowledge that. And I hope that anyone who wants to encourage us, support us, like please think of ways to help us. Yeah.
Here's a very specific question. Why do you think the SARS-CoV-2 could not have come from the Mojang mine?
I'm not saying that it could not have come from Mojang mine. I'm saying that the strains that we know collected from the Mojang mine up to 2015 are not the direct precursor of SARS-CoV-2. That doesn't mean, though, that no other SARS-2 viruses were collected from that mine or the region around that mine or any of the 7 countries shipping pathogen samples up to Wuhan.
And there's another question from the chat. There's been congressional investigation, if you've mentioned. Are there actions that Congress can take, do you think, that would be helpful? Are there a couple that you would recommend?
Well, the first thing, based on all these recently FOIA and leaked documents, is that it looks like the EcoHealth Alliance knows a lot more about the type of research that was happening in Wuhan than they let on. So maybe we should see those documents. So, okay, maybe if you're worried about some sensitive information, we should definitely get a formal investigation going, one that is bipartisan, credible, has scientists on all sides of the issue looking into it, and let that investigative team take a look at the documents. You don't have to post it on the front page of the New York Times, is what I'm saying.
And do you think there are, another paraphrased question from the chat, do you think there are actions that we can take to encourage dialogue with China about this open dialogue?
I think that some of these have to be systemic and structural. The way that we publish papers, the way that we accept like scientific currency in the future, is that one example maybe is that journals should all band together internationally and say, we're not going to publish any more papers where we see that pathogen sequences have been held back for 3 to 5 years. Like there should be a new international protocol. As soon as a sequence is derived off the sequencer, the raw data just goes straight into the cloud. So anyone trying to publish a sequence that was like they kept in a repository for years and years, no, we won't accept that anymore. So there are ways to build in transparency into the scientific process.
And do you think that's, just to follow up, do you think that's the job of the journal editors or who?
I'd say that I think it's everyone's job. Like we can't say, oh, it's the journal editor's job, because there's no incentive for them to do this. We can't say that it's the author's job, because the authors don't want to do it. No one wants more paperwork and more regulations on their works. We all have to do this together. And that's why I think it's so helpful if we could have a new moratorium, and not just one that has like closed door with a few scientists in there, like every public stakeholder also needs to be there.
Here's a question. Lacking cooperation from the Chinese government, if that's the way it goes, what would be the best evidence that we could hope to stumble upon?
So again, I don't feel like we're particularly desperate right now. There's so many sources of information we haven't even tapped into that I don't think we should feel hopeless and pin all hopes on a whistleblower. Although I think that there would be eventually a whistleblower. It's like many of these other lab outbreaks that have been covered up over the years, and even natural outbreaks have been covered up. So eventually somebody who knows something might say something when they feel safe enough, when they don't feel like they're going to be sent to the firing squad or their co-workers will be thrown in prison and tortured. So especially in this world, in this era, like this, everything is recorded. Your emails are recorded, like your phone is on you all the time. Like you can record things, you can write things and throw it into a hard drive or something. Like there's so many ways to correct. You're no longer chiseling things into rock or like, saving a piece of dried up paper somewhere. Like there's so many ways to record information and evidence.
Yes. What if you, here's a question. Let me just check. Let me just pull this back up.
Mm-hmm.
Seeing as how an accident in one country has the capability to affect the globe, what kind of international coordination and guidelines on BSL requirements should be implemented?
Yeah, that's a very insightful question. It's because there are just dozens of labs around the world doing this type of research, and each one has their own standards. So there are guidelines that are shared, but there's no enforcement. So there should be a pandemic treaty, a new one that's ideally not enforced by the World Health Organization, because we have seen now that they have zero powers to enforce such a treaty. And there should be clear penalties for not allowing people to come in and investigate when the outbreak happens. So many experts, they'll tell you that the inability to send people in the early days of the outbreak is what is causing so much trouble today. So all this information about whether it can spread human to human, whether it can spread pre-symptomatically, whether it's airborne, like this information had to be slowly leaked. Like each country almost had to do their own study to believe the data. So this should not be the way the next pandemic unfolds. We need to have a pandemic treaty.
That's great. Well, Alina, thank you so much. If you could stop sharing for one moment,
yeah.
we'll put up the final slide. I want to point out to the participants that we have put the link to your book back into the chat for folks who are interested. I can't thank you enough and wish you all the luck on your book launch tomorrow. We'll be watching that closely and reading your book. And I'm going to also point us towards upcoming talks in the series. Amy Abernathy, who is the president of clinical study platforms at Verily, but also the founder of Flatiron and former FDA CIO and deputy, also the deputy director of the FDA, Todd Golub, back from Alina's home institution, director of the Broad, Rob Knight, who's perhaps the foremost voice on the microbiome in the United States, Vinny Prasad, a skeptic and provocateur and evidence-based medicine advocate and leader, and Christina Farr, now in OMERS Ventures, but formerly a top reporter for health and health tech, a unique niche that she really helped to create for CNBC. You can find the events, past events, and future events at www.chip.org. Feel free to learn more about us there. Alina, again, thank you so much for a spectacular afternoon with you.
Thanks for having me. I'm really honored again to be here and to be able to speak about this really important topic.
Thank you.